My name is Michaela Kerber, and today I'm going to tell you a little bit about my summer research work supervised by Dr. Arunika Gunawardena and Dr. Zhenyu Chang. The lace plant, or Ponegate madagascariensis, is an aquatic monocot that is considered an emerging model organism for describing programmed cell death in plants for three reasons. First, it undergoes predictable perforation formation via developmental programmed cell death. Second, its thin leaves allow for long-term live cell imaging. Finally, the plants are able to be grown in sterile cultures for pharmaceutical experiments. The lace plant is propagated in the Gunawardena lab using in vitro asexual propagation. The method used for propagation is called tissue culturing, which is a fundamental biotechnological technique that usually involves the growth of plant cells or tissue on a nutrient medium within a controlled, sterile environment. Despite use of a laminar flow hood to maintain sterility, the sterile conditions in the lace plant's aquatic environment are easily compromised, as it is optimal for both the growth of plants and unwanted microorganisms. Contaminated cultivars result in economic loss for the lab and underscore the need for a simplistic procedure that eliminates contamination in our lace plant cultivars. With this in mind, the goal of my work was to identify the 4 common contaminants found in the lace plant cultivar and use that to develop a simple protocol for sterilization. Identification of these contaminants began with growing and isolating colonies from the material that was found in these cultures. The resulting colonies underwent DNA extraction and Sanger sequencing, then a pair of computer programs were used to analyze and identify the best genetic matches for each sample. Results showed that a wide variety of fungal and bacterial species were present in the contaminated cultivar, and many of these species were isolated from the same plants every time. The 3 sterilization protocols that we tested were a surface sterilization protocol, a broad-spectrum biocide treatment protocol, and a combination protocol of the above 2 methods. We began with a surface sterilization protocol, where contaminated cultures were pretreated with bleach and grown in an untreated media. We then performed the broad-spectrum biocide protocol, where contaminated cultures were grown in media containing a variety of concentrations of biocide to determine the best concentration for sterilization. We finally combined both methods by pre-treating with bleach and then growing the plants in the best concentration for a month. Results showed that the first 2 protocols were able to eliminate contamination in up to 75% of the plants tested. In all, we found that the combination treatment was the most successful, eliminating contamination in up to 93% of the plants tested. In summary, the combination methodology that was created through identification of the contaminants in the cultivar resulted in the most successful sterilization. Future work will involve performing more replicates of all 3 methodologies. I would like to quickly thank both of my supervisors and all the members of both labs that helped me this summer on this work. Here are my references. Thank you so much for listening today.